Okay, this is part 2 of the cellular respiration notes. I know you're all very excited, but please try to contain yourself. We're going to talk about Krebs cycle and electron transport chain. So first let's start with the Krebs cycle. Oh boy. Krebs cycle is also known as the citric acid cycle, and you'll see this if you look it up in your book or Wikipedia or most other places. It is a series of chemical reactions that take place in the matrix of the mitochondrion. It makes ATP, most importantly electron carriers, and also produce CO2, which is basically a waste product. Next term we need to define is acetyl-CoA. Pretty simple. Acetyl-CoA is a 2-carbon molecule that forms from pyruvates that come from glycolysis that start the Krebs cycle. I put start in quotation marks because there's really no start of a cycle, but that's the best place to talk about where it starts. Next molecule is OAA, and this stands for oxaloacetic acid, but it's one of those words that you don't want to try to remember. But very simply, it is a 4-carbon molecule that stays in the Krebs cycle so I can keep going. This is much like RUBP that we saw in photosynthesis. Dark reaction. Citric acid is a 5-carbon molecule that is actually created in the Krebs cycle, why it's also known as citric acid cycle. It's created in the Krebs cycle, and it is actually the molecule that's going to be broken down a couple times to cause electron carriers to be made, ATP and also CO2. Before we get into the Krebs cycle diagram, I just want to go back to the rest of this, and that is number 6, explaining the role of electron carriers. And we've already seen these guys in photosynthesis, so it should be kind of a no-brainer. They provide electrons for the electron transport chain. And the electron transport chain is taking place on the inner membrane. Also known as the cristae. So taking place on the inner membrane is the ETC. And that's where these electron carriers are going to get used. Remember, just like in photosynthesis, to write them with 2 little dots right there to represent the electrons. And also right there. We have 2 different electron carriers this time. They're going to come in at different locations, but they're both doing the same thing. We don't care what the letters stand for. All we care is that they are carrying electrons to the electron transport chain, which kind of should be a no-brainer because electron transport chain tells you pretty much what it is doing and what it needs. So going back to the Krebs cycle, hopefully this computer cooperates. I'm going to take these things out of the way, just a couple of these things out of the way so we get a better look at the Krebs cycle. And up here I've got a picture of a mitochondrion, and that's where everything's happening for this unit, in and around mitochondria. So keep in mind we're talking about Krebs cycle taking place in the matrix of the mitochondria, and that is the very inside. And something else we need to talk about here to get a good idea of what's actually happening. Right before the Krebs cycle actually begins, the pyruvates that are coming from glycolysis actually get turned into 2 P-gals, so 3-carbon pyruvates get turned into 2 P-gals, but still 3-carbon molecules. These guys are eventually going to hook up with a coenzyme A spit off one of the carbons, and the other two carbons are going to form with coenzyme A to become acetyl-CoA, and that's this guy right here. CoA, so put a little space between there, or you can put a hyphen, whatever floats your boat. So acetyl-CoA is actually the molecule that, again, quote-unquote, starts the Krebs cycle. This is very similar to CO2 that was coming into the Calvin cycle, or dark reactions in photosynthesis. Again, it's a cycle, it keeps going, but there are carbons that have to come in to make sure that that cycle continues as a cycle. So the first thing we're going to label besides this guy here, acetyl-CoA, is our other molecule that we talked about right over here, citric acid, and that is this guy right here. Citric acid, you can see there is a 6-carbon molecule. That is the formation of acetyl-CoA and this molecule that stays in that we'll get to at the end. First thing that happens, actually we're going to break this, it says here, summarize in 3 steps. So let's call this our first kind of step of the Krebs cycle, and that is formation of citric acid. So if you want to just off to the side somewhere, Krebs cycle, steps. Formation of citric acid, Ca, I'm abbreviating, citric acid. So that's this part right here. Again, we'll talk about this 4-carbon molecule in just a second. So first thing that happens here, besides a creation of a molecule, is something that we've kind of discussed in photosynthesis when we talked about the transferring of electrons and the recycling of those molecules that actually do the transferring. So the first thing we're going to talk about is how NAD plus is used here. It's actually something that gets sent off back to glycolysis. How NAD plus gets made into NADH. And again, NADH, you might want to put a dot in there between the D and the H. Remember, that's the electron carriers that are getting sent to electron transport chain. Something else that's getting spit out here, a little bit less important for us, is CO2, which is just a gas waste product. But you can see there was 6 carbons here. Maybe you want to label this citric acid, 6-carbon molecule. Breaks down into a 5-carbon molecule that we don't care the name of. And one of the carbons comes off as CO2, a waste product. When you exhale CO2, so that's one way to think about this. This is going to happen a couple times, where NAD plus comes in, picks up electrons, and becomes NADH. And again, you probably want to put your little dots there. 2 electrons to help you remember what the purpose of that molecule is. And again, notice what happens here. There's a carbon that comes off. Another carbon is going to come off as CO2. So it's just making and breaking molecules, just like we saw in photosynthesis, dark reactions of photosynthesis. So there's a carbon dioxide also coming off in both of those locations of the Krebs cycle. Scoot up a little bit. Next thing that happens, and you can see kind of at the same time here, there is a formation of an ATP molecule, and the Krebs cycle is only going to make a couple of these. And again, remember, ATP is our energy molecule, so maybe a way to remember that. Put the little fireworks sign around it. And so we're going to call this next phase down here, let's label these 1 and 2. Step 2 we're going to call the breaking or the using, maybe would be a better way, breaking citric acid a couple times to get electron carriers, ATP, and least importantly, CO2, a waste product. Again, this is just a gas. It's just a carbon coming off of the molecule, 5-carbon molecule here, and off of a 6-carbon molecule up there, coming off as a gas. Step 3, just kind of draw those lines into your diagram. Step 3 we're going to call regeneration of OAA. And that is the 4-carbon molecule that keeps the cycle going. The dark reactions we saw, 5-carbon molecule that keep the cycle going. This time we have a 4-carbon molecule called OAA. And a couple things that are happening here. FAD plus is coming in to make FADH2, again, just electron carriers. So we're trying to put a couple dots there. And also here, making more electron carriers, but in this case, NADH. And that is the Krebs cycle. So 3 parts. Formation of citric acid using OAA, this guy. Now we can label it since we've talked about it. A 4-carbon plus a 2-carbon to make a 6-carbon. OAA plus acetyl-CoA to make citric acid. A lot of A's in there. OAA, acetyl-CoA, and Ca for citric acid. Then the citric acid gets broken down a couple times, making electron carriers and ATP, also a waste product, CO2. And then these guys are going to stay in to allow OAA to reform so that the Krebs cycle can continue. And before we go on to electron transport chain, I'm going to stop this video so we don't run out of time on YouTube.